So, hello everyone. So, I thought it's a good idea to share with you the project that we're working with the research team. So, it's about variability and underpotency of cholecalciferol content of commercial vitamin D preparations. So, we talked in the last 2 weeks about the vitamin D and the prevalence of vitamin D. Two-thirds of Canadians have an adequate serum 25-hydroxyvitamin D, with the mean levels of 20 in the winter time and 25 in the summertime. And as you mentioned, Dr. Bazikin, that the global prevalence of vitamin D deficiency, 64% of women had a serum a level below 30 nanogram per millimeter. And we talk about the vitamin D resources. We saw that sunlight exposure that's unreliable, and that diet sources is minimal or not meeting the requirement. So Institute of Medicine recommend dose of 400 to 800 per day for all adults above age of 50, not osteoporotic patient. And the Endocrine Society recommends 600 to 800 for all adults above age of 50. Majority of patients receive their vitamin D supplements in form of vitamin D over-the-counter preparation. So our research question, why some patients do not have the expected increment in their vitamin D level when we are giving them the usual amount of supplement? Could one reason for this non-response be inconsistent amount of vitamin D preparation? Of course, after excluding the compliance and malabsorption issues. It has been a hot topic in the news, and this one, supplements, are we getting what we promised? They study vitamin C, and where they report that a little bit of vitamins and a lot of sugar. None of these products contain the amount claimed by the company. And Health Canada acknowledged their weak evidence for approving herbal vitamin D supplements. Within 9 days, they get the approval of vitamin D supplements. They have regulation. I'm not sure if you share the same regulation or not, but 1,000 or more it's considered as ethical pharmaceutical, and less than 1,000 or less considered as over-the-counter with the potency required from 80 to 120. So our study aims to evaluate what's the content of vitamin D3 in both over-the-counter and the prescription sources of vitamin D in Vancouver. So what we did, we collect 23 vitamin D products from the pharmacies and health food stores, and I will not go into details of methods, but the tablets were pulverized, and we added antenna standard, vortex, then centrifuge, and we use the methanol extraction method, and we use also the high performance liquid chromatography mass spec technique for identification of vitamin D. And the assay was validated by repeating the test 5 times. Recovery and extraction efficiency were evaluated also, and also the reproducibility. We didn't test for the stability because it was mentioned before in many studies that vitamin D is a quite stable vitamin. It's coming to the results. So 400 IU, 10 of them, as you see here in the figure, it was large variable, and with a variability of from 8 to 120%. And 80% of the product was below 80% of the label amount. Yes. And not only 400, but also 1,000. And the range was from 9 to 114%, with a potency of 80% of the products below 80% of the label amount. We thought that's only for the over-the-counter medication, but also the pharmaceutical. So we test the 50,000, it was reasonable, but the 10,000, 2 of them that we tested, 10 and 12%. And just to point that the 50,000 was from US source. It's the only one from US. And reviewing the literature back, so there was LeBlanc that was study that was published in 2013, the US study, methanol extraction method they used, and the HPLC used, not mass spec that we used in our study. They test for 55 over-the-counter with a compounded also multivitamin products. With the potency was ranging from 9 to 146. And half of the tablets meet the US standards, which is 90 to 110. And there was a letter to the editor from Agriculture Research Service of U.S. Department of Agriculture that in their pilot study showed the same variability, and they recommend to use internal standard and certified references material just with the mass spec technique that we use in our study to rule out the interfering compounds. Another study from New Zealand, they use methanol extraction as we did with HPLC alone, and they examined 13 samples with the potency again from 8 to 200, and the prescription was from 90. Actually, the prescription was within the acceptable range in this study. So could one reason for this inadequate amount of the vitamin D that we test, or instability of vitamin D, although it was mentioned previously that's quite stable vitamin, deterioration of the supplements after manufacture date, one possibility, or incomplete extraction of vitamin D3. So we use methanol extraction, does other solvent can extract more. And also we hear this the debate regarding the effectiveness of vitamin D supplements preventing fracture. So underpotency, is it one reason for that? Adding to this controversy is about the pre-25 hydroxy testing and also binding globulins that might complicate the whole picture also. The limitation for our study, it was random sampling, and we didn't evaluate batch-to-batch variability, and we used methanol extraction method. So more non-polar fat soluble like hexane can extract more, we don't know. And also the bioavailability of the study of vitamin D was not tested. So in conclusion, unlike other vitamins, vitamin D, we commonly rely on the vitamin D to treat some serious medical diseases, talking about rickets, osteomalacia, or osteoporosis. With this large variability in vitamin D supplement potency was a significant concern, of course, from both public health standpoint and individual therapeutic perspective. The future direction, 3 weeks ago we got the news from one of the Health Canada staff that is working with them, Steve Brooks, that he's interested to proceed with reproduce our data with different lab and different way of extraction. They will do hexane extraction that I think it will be valuable. And also maybe bioavailability and what's the clinical importance of this finding. So I think it's very important. So questions or comments?